Ruth, thanks for doing this interview. I would just love to hear your impactful research and the most exciting story you want to share with the public about why your research matters and what is the recent big news.
Okay, great. Well,
Yeah.
my research is all about using the data that we have to get to the evidence that will help people to make good decisions. That's very broad. So I'll just talk about one particular area that has been a big focus of my work, which is the area of cancer screening and early detection. And this is a really exciting time for this field because we seem to have cracked the code for use blood tests to detect a whole range of cancers. Previously, we only really had blood tests for prostate cancer and ovary cancer. But now I think it looks like we will be able to use blood tests to test for a range of cancers, including cancers that don't have screening tests. And some of these blood tests can test many cancers at one time. One blood test can detect one of many cancers. So this is really very exciting for people who I think rightly feel that early detection is one of the major tools we have for cancer control. But there's now, used to be in the past, very few screening tests to evaluate, and now there's going to be a lot of tests to evaluate. So a whole machinery for test evaluation, including how we do screening trials, now has to be reassessed so that it is nimble enough for this new world of many tests. And so we want to make good decisions. We don't want to adopt tests that are not actually useful or tests that can bring harms that we don't fully understand. So we need to understand harms and benefits of the test before we introduce them to the public. But there's a lot of momentum and pressure to get these tests out to the public quickly. So the question that I'm excited about is how can we evaluate these tests reliably, but in a short time frame? And I mean, this is a problem that has come up in other areas. I mean, in treatment trials, surrogate endpoints are used to make the trials shorter, or in vaccine trials. Also, there's a whole methodology for surrogate endpoints. So how can we learn from these different types of trials, take the lessons from these trials into screening, which is different, to try to also, in a reliable way, shortcut these screening trials? So I'm really excited about that challenge right now because screening is different from any of these other applications. But I think that we are very compelled to try to find a solution so that we don't take, 10 or 15 years to evaluate new cancer screening tests.
Wow, that's very exciting to hear. So I'm particularly curious about what breakthrough led to this screening test to become effective for other types of cancer besides ovarian and prostate cancer. Yeah.
Yeah, that's a great question. And really, it's the science of liquid biopsy. Now, it's not new that we understood that some proteins, some products of cancer, even solid tumors, would circulate. That's how we got to having tests like PSA, or CA125. And these are proteins that are secreted. They're also secreted by healthy tissue, but they're secreted more by cancer. But after that, people started to recognize that actually there were circulating tumor cells, that you could find tumor cells circulating, and people who are interested in metastasis, recognize that this might be part of the process. But what I think really opened it up was the recognition that it's not whole cells that we might find in the circulation, but fragments of DNA that we might find either floating freely or in extracellular vesicles, exosomes, that because cancer cells have a high turnover and there's a lot of cell death, then these products come into the bloodstream. So there's not only circulating DNA. I mean, there's circulating RNA. There's less stable. There's all kinds of immune system perturbations that you can see. But I think what people realized was that they might be able to find these fragments of DNA, and they're from healthy cells and tumor cells. But tumor cells have specific fragments that are different from the fragments from healthy cells. And so interrogating these fragments of DNA, started to give people a window into the cancer process. And this actually happened really in the part of the continuum that's really after diagnosis. And there's a big area of research in minimal residual disease and circulating tumor DNA for identifying early recurrence and evolving phenotypes of the cancer post-diagnosis. But then people recognize these particles, these fragments are present even before diagnosis. And this then led to the notion of using these fragments for early detection tests.
I see. Thanks for the explanation. So regarding the shortcut you hope to achieve for the screening trials, what are the strategies that can make it work?
Well, the question is, can we, there's two things. One is shortcut and also reduce the size of these trials, because there tend to be tens of thousands of people, and over 10, 15 years, it's prohibitive. So there's a couple of shortcuts that people are investigating. To reduce the size of the trial, there's some interesting innovative design ideas that capitalize on the fact that if you have a biomarker, you can store it. Right. In the imaging test, you can do it on, you don't do it on healthy people. Biomarker you can collect from healthy people. So there's nice ideas for design to reduce the size of the trials. Then to shorten the trials, the key avenue is to find some sort of short-term incidence-based surrogate endpoint for the usual endpoint for these trials, which is mortality. So the one that's been talked about the most is, instead of looking at deaths from cancer in these trials, can we look at how many late-stage diagnoses are there in the screen and the control group? Does the screening reduce late-stage diagnosis? So late-stage diagnosis as a potential surrogate endpoint for mortality. And people are trying to investigate this. But there's other possible surrogate endpoints that we could consider. But that's the one that's most popular, and it's the one that's easiest to explain. And the last way that I'll say that people are thinking about shortening trials is, in some cases, not to do the trials at all. If we have a trial already for a set of cancers, and we have the surrogate endpoint, maybe also the mortality endpoint, and now we have a new test for a similar set of cancers or a subset of those cancers, and we understand very well the differences in the diagnostic performance of the two tests, might we be able to use a modeling approach to investigate the new test, to do like a virtual trial of the new test? So this is very germane to our work in modeling. And we are kind of on a bit of a campaign to rethink which trials should we do. And because there's going to be lots of tests, we won't be able to do them all the trials, but can we do big trials of the tests that will enable us to learn enough so that we can model the next test? That actually the goal of the trial should be to learn the model, also to learn the efficacy, but to learn the model so that we don't have to do future trials. Kind of a revolutionary idea, but
I see.
Somewhat self-serving to propose. But we think this might help in some cases to even not even have to do trials. Yeah.
Wow. Is this also related to this digital twin idea?
Okay, so the digital twin idea is not the same. I'm saying we'll do a digital trial. The digital twin idea is more in clinical, let me think, clinical, I don't know, maybe there's a connection, but it's more, I'd say, I've seen it more in clinical outcomes of a certain intervention, usually treatment, but I guess you could do it for screening. Yes, you could do a digital twin if there is a screening test being used in practice, which can happen with screening tests. They get into practice before you finish the trial. Yes, you could. But it's not a trial. It would be like a trial in the sense that you're trying to find a control group from your observational data that looks like your screen group, people who are getting screened in observational data, and you're trying to create, that's actually a great question. I didn't see the connection until I'm talking about it. But you're trying to create a control group in an observational data setting so that you can learn the efficacy of an intervention, which could be a screening test. In that setting, you could learn the efficacy of the intervention by kind of doing an in silico trial. Yeah.
Trial. I see. So regarding the model, which sounds very exciting and is most related to our program because we are computational researchers, right? So how many variables do you want to incorporate into this model? And are you going with more like this AI route or more like the parametric transparent modeling route?
No, it's just great that you asked, but well, I've always done this in a very mechanistic way. So I would start with that. And, these models are, there's two models that we need. One is a model, so one might use AI more than the other. One model is using the patterns of screen and interval detection to learn the natural history of the cancers. This is a very kind of old methodology that has been used in HIV, chronic disease, and in infectious and chronic diseases to learn about the latency of the cancer, that kind of unobserved natural history before diagnosis. That's one model. Another model that we need, though, is a reliable model that links the surrogate endpoints to the true endpoint. So it's a predictive model for the true endpoint given the surrogate endpoint. And to the extent that that model might incorporate other features at the time of incidence, at the time of diagnosis, that could turn into, certainly as a predictive model. Whether would need AI to do it, I don't know yet. But what we know from the vaccine literature is that in vaccine trials, and people at Fred Hutch were very instrumental in this, in the vaccine and vaccine trials network and the prevention trials network. Peter Gilbert and all those folks did this really great work developing causal predictive models to predict the true endpoint in the vaccine trial, which is incidence of disease, from the surrogate endpoint, which is some kind of antibody titer. Neutralizing antibody titer, for example. And even those models were not AI based. It was more important to show that they were causal, to get them to be, causal, so that you really could transport to new trials the predictive outcome from the surrogate endpoint. So, there are issues beyond, you could talk about AI because it's a predictive model, but what's more important is that you can trust it from a causal perspective.
Oh, wow. This is so cool to hear. And
I'm so glad you think it's interesting. I think it's interesting. We always think our own work is interesting. It's great when we, when we can communicate in an interesting enough way to other people.
Yeah, especially its relevance and importance are not to argue, right? It's very obvious that if you can shorten the trials, then more tests can be available to the public in a much shorter time frame.
Exactly. And there's a lot of optimism in this regard. This really, in the treatment trials, really grew out of activism from the HIV advocates. But here, the push is coming from the companies making the tests. And they're all ready to do short trials. In fact, the first trial of a multi-cancer screening test just reported results, and it was a 3-year trial. We've never had a 3-year trial in screening, but it ended at late stage incidence. The company just used it. But, whether that was valid indicator of effect on mortality had not been worked through. But the company was perfectly happy to do that because they needed a short-term trial. So what's interesting is that the activism is more coming from the producers of the tests as opposed to, from patients. But everyone thinks early detection is a good idea. So, I expect it will continue to, spread among patients as well.
Great. So if I want you to give some advice to our younger generation, especially students like undergraduate students or graduate students who are interested in your field of research, what kind of expertise should they learn in this current era, especially with AI? Like you can learn things in very different way now.
Yeah, I understand that. I think that in the natural history space, I really think that the methods have evolved tremendously. But what I'm seeing in the space of natural history modeling is that Bayesian approaches are really the way to go here.
Mm,
And so I think it'd be really important for them to sort of learn that.
mm,
And how it applies in this case, using Markov chain Monte Carlo. There's a lot of issues of identifiability in these models. So using Bayesian methods with well-specified priors can help you with those identifiability problems and the complex modeling. And then on the predictive side, I do think that we will need causal models. So I think that causal methods are very important. And to me, that's the intersection of causal with AI. So predictive methods that can, be very accurate, but also have a causal interpretation. I think that's a really interesting growing area of our field, is this interface between the rigorous, mechanistic, reliable statistical methodology and then the looser kind of AI approaches that often many people don't even understand, but to put them together at the interface. And I think that our program actually is very well equipped to be at this interface. And I think that the Hutch in general, if we can get good collaboration going between the different quantitative sciences, some of which are more AI heavy, like the IR data science, IRC maybe, and then our program, which is, coming from traditional statistics, that collaboration could be a real advantage for Fred Hutch.
Thank you so much, Ruth. Yeah, this is so informative for our public and also for people who are interested in statistical and computational research. I greatly appreciate your time in introducing your research and your insights to us.
Thank you for the opportunity. Great questions. Yeah,
Thank
it was
you.
a pleasure.